The Autism Research Institute relies on the generosity of donors like you to make this webcast possible. If you enjoy this presentation, please consider making a donation. Thank you.
Hello, this is Dr. Andy Zimmerman. I'm very glad to be here and glad you joined us. I'd like to talk to you today about the subject of neuroinflammation in autism, and I've titled this Neuroinflammation or Immune Dysregulation. That is the question, and we'll proceed here. This is my view of autism over the years. It's been a while that I've been working in this field, and it has often seemed to me like the blind man and the elephant all looking at different aspects, and everyone finding his or her own area of interest, and of course looking at each part of the elephant, finding what we think are answers, and in the long run, I think, are coming together slowly into a more coherent understanding of this very interesting, very puzzling, and of course heterogeneous disorder. We all want to look at what interests us most, and there are so many things in the field of autism that you can concentrate on. You know that we know that the immune system plays an important role in autism. It's been known since the early 70s, and more and more information has become available. I'd like to talk to you about that today. But we won't forget that the GI tract is involved, sleep is involved, synaptic function is involved. Prenatal infection was the first thing that gave us biological clues to autism. And of course that was recognized by ARIES founder, Dr. Bernard Rimland, who made great contributions to the field. And we will talk later about mitochondria in autism. So this is my outline today. We'll first review some of the immune findings in autism spectrum disorder, or ASD. I'll talk to some about immunogenetics and HLA. And we'll go on and talk about autoantibodies found in maternal and child sera. Question is whether they're causes or effects. And I'll talk about microglial activation and the question of whether it's neurotoxic or neuroprotective. Then I'd like to talk more generally about neuroglial functions. In other words, not just neurons and microglia, but also astrocytes, and talk about their interactions and try to think about a broader perspective of the problems in autism at the cellular level and how microglia might be part of that greater picture and the immune system. Then I'd like to talk about prenatal insults and reprogramming and the question of whether autism may represent a form of arrested development. Then we'll talk about immunomodulatory therapies and questions about what treatment should represent and at what level. And finally, we'll talk about cellular metabolic and synaptic dysfunction, and we'll mention the fever effect and a somewhat different approach to therapy. A very excellent paper is this by Zietz and Rennert in 2011. In this paper, they talk about gene expression in autism. Now, we know that enormous amounts of effort have gone into the genetics of autism and gene discovery. And some of those studies have implicated immune system genes. But what Zietz and Rennert emphasize is that it's the expression profiles of different genes that identify specific nodes in these interacting networks that are important. And in two distinct networks, they find specific immune genes. Here's a very important regulatory gene called NF-kappa B. Others are related to TNF here in network number 2, NF-kappa B. Tumor necrosis factor alpha is a very important one and increased in autism. TGF-beta is another. We're now beginning to recognize that it may not be the genes themselves, although certainly mutations and polymorphisms are important in autism. It may be the way the genes are expressed, and that gets into an area we call epigenetics. It may be the epigenetics and the way gene expression is controlled in autism more than the genes themselves. Now, early on, as I mentioned, in the early 70s, it was first recognized that there were some families who had a lot of autoimmune disorders in the families. And this was recognized by John Money, a psychiatrist at Johns Hopkins. And later we went on, when I was working in Tennessee, to look at families in our cohort there for their HLA types. HLA are the immune system determinants that are coded for, and certain HLA types are increased in autoimmune disorders, and we found also in autism. This was also reported subsequently by other groups. We found an increase of the HLA type DR4, which is also increased in rheumatoid arthritis, although we did not find this when we looked at the AGREE sample from around the country. We felt that this might represent a regional variation, and subsequently other authors have found differences in different areas as well, although a large-scale study in Scandinavia has corroborated the association with autoimmune disorders. So we know that there's something genetic about the way immune systems are controlled in families that has a role in autism. Now, an enormous number of, a few hundred papers are now available looking at various elements of the immune system. Here is a list of cytokines, mostly from Paul Ashwood and his group at the Mind Institute at UC Davis, finding a number of elevations in the peripheral immune system, in the blood, in children with autism, elevated levels of IL-1B, IL-6, IL-8, and so on. And down the list you can find almost any element of the immune system and cytokines that has been looked at in different cohorts of children with autism that show either elevations or in many cases actually decreases in in the amount of material that you're looking at. And this has left us in a quandary in many respects as to what do all these different changes mean. Here we're looking at cytokines and chemokines. Chemokines are small proteins that are produced by the immune system. And here again, we see many different authors have found different changes in the immune system that are puzzling. And in most cases, they indicate a lower level of functioning, a less able system, but in many others, they represent an increase in the activation of the immune system. Here is an excellent paper from 2010 by Paul Ashwood's group showing T cells in autism, when stimulated, produce increased amounts of TNF-alpha, but another IL-12P40 was decreased relative to typically developing controls. In general, there's an increase in pro-inflammatory or Th1 cytokines, and they found that they were associated with greater impairments in the core features of ASD, as well as aberrant behaviors. In contrast, production of GM-CSF and Th2 cytokines, the less inflammatory, were associated with better cognitive and adaptive function. So this and other clues seem to indicate that there is an association between immune dysfunction and clinical attributes in autism. And this has not been studied nearly as much as we hope it will be in the future. Here is a very interesting recent finding by two groups, Torres et al. And by Guarini's group, looking at natural killer lymphocytes. Now, NK cells are very important because they are an innate, immediate reactive cell when they encounter an infection or a difference presented to the immune system. They don't have to be primed by by previous experience, and they rely on antigens or proteins on their surface called the killer inhibitory receptor, and this varies in autism spectrum disorder, and they find that this is associated with decrease in NK function because of differences in the protein. So this is very exciting. It was first reported by Torres et al. And confirmed by Guarini. And the interesting thing here is that the Guarini's group found that this was present, this problem is present both in the children with autism as well as their mothers. And this might be one of the links between the autism, between the problem with autoimmune disorders in the family and with expression of the immune system abnormalities in the children. But we have a long way to go to understand this. These are exciting clues. Now this is from a paper by Hogberg et al. In the Annals of Neurology in 2012, just showing the phases of inflammation after an acute sterile insult. You don't need bacteria to get this insult. You can have it from any kind of toxic exposure or an injury. So there are the point here is that there are phases of different immune response, parts of the immune response that occur from different cell types in the immune system, and that there is a timed regulation of these changes. Now here we're only looking at 14 days, but you might imagine that if you have an abnormality in your immune system and the way it is regulated from one part to another, that this problem of persistent reactivity may be persistent way beyond this, and more likely than not for a lifetime. And we'll go into this later, because the changes in the immune system, particularly in the brain in autism, are lifelong as far as we can tell. They may come and go and they may cycle, but they do appear to be lifelong. Now I'd like to shift gears and talk about maternal and child antibodies. And as you're probably aware, a lot of work is being done by Judy Vanderwater and her group at also at UC Davis. And it has now become clear that 10 or 15% of mothers who have children with autism carry antibodies that are directed to the fetal brain, specifically the fetal brain, not the adult brain. And these were first described in 3 different types, the 73 kilodalton. These are bands on a gel, portions of the antibodies are found at different molecular weights at 37, 39, and 73 kilodaltons. Much work has gone into this, and many mothers have given blood samples. We showed with Harvey Singer and when I was in Baltimore, that these antibodies, human antibodies, could be transferred to mice, pregnant mice, and the offspring would had abnormal activity levels and reactivity, and this was very suggestive of hyperactivity and other abnormal behaviors. Of course, there is no perfect mouse model of autism, but they were distinctly different than the offspring of mothers who had received just saltwater injections. And on the left here, you can see, sorry for the glitch there, that these fluorescent antibodies show up microglia activation. Now, microglia are the resident immune cells of the brain, and they were activated by this process, giving the human maternal antibodies to the mouse mothers. And these were the controls in the middle, and on the right was the salt water injected. So, and this subsequently was also done by the group at UC Davis in monkeys. They transferred maternal antibodies, human maternal antibodies, to pregnant monkeys and looked very carefully at the offspring, and they had many autistic behaviors. It's been well done. Now, this is from a recent paper by Judy Vanderwater's group that demonstrates their finding of specific targets, antigenic targets, to which these antibodies are directed. And they are listed here, YBX1 and so on. these different proteins are responsible for specific functions in the developing brain. In this case, STIP1 forms neurites. Cytokine is important for dendritic branching, and so on. So these antibodies, the maternal antibodies, and what they're directed to are now being defined, and we're in the process, this group and others are in the process of determining what these antibodies are like on a larger scale, what their predictability is, and what the risk is for a woman who is carrying these antibodies. One important question, or many important questions, but one very important question is if a mother has these antibodies, what is the risk for the next child? How are these antibodies generated? When are they generated? What generates them? Is it exposure to a previous pregnancy, and so on? There appears to be no specific birth order defect. In other words, if a mother has these, what is the risk for subsequent children? Because so far that it does not appear that every child is susceptible. And this suggests that there might be a susceptibility factor in that child, him or herself. Autoantibodies have also been described in children. Here is a description of antibodies directed to Golgi cells in the cerebellum. You can see here that are staining. This is in the human cerebellum, showing antibodies directed to Golgi cells. What these mean isn't clear. When we looked at this, we couldn't find a specific effect of autoantibodies from children compared to controls. But this is still an open question as to whether a child may be making antibodies directed to the brain in autism. A big question we had 15 years ago now was whether the abnormalities being described in the peripheral immune system that you can draw from the blood were effective or had any corollary in problems in the nervous system. Because, as you know, the two are separated by what's called the blood-brain barrier. And the blood-brain barrier protects the brain, the nervous system, from invasion by antibodies and by cells from the immune system. Although there are some that do cross, in general, they don't. Now, this cell I mentioned, called the microglial cell, is a specialized cell that's actually derived from bone marrow, which has been proven. And it comes from the bone marrow lineage and ends up in the brain and makes up a substantial number of about 10 to 15% of cells in the brain. And it's been recognized for many years, although its functions have not been well described. We do know that it becomes activated, and I apologize for this arrow. It becomes activated in the presence of insults to the nervous system, and when it's activated, it can have what we call classical activation, in which it secretes pro-inflammatory cytokines, these small proteins that have important effects on the cells in the nervous system. Or it can also produce what we call alternative activation, which is actually anti-inflammatory. it controls inflammation. And then there is a function called acquired deactivation as well. And it is not entirely clear, unless you are looking at an experimental model, whether microglial activation in the brain is pro-inflammatory or anti-inflammatory. The appearance of the microglial cell changes with its state of activation. Here you can see changes in the cells with activation or alternative activation, and with immunosuppression. Now, with Diana Vargas and Carlos Pardo, we published in 2005 that microglia are activated in the brains post-mortem from people with autism from 5 to 44 years of age. And this has been subsequently corroborated in various ways by other groups. It's not just the microglia that are activated, it's the astroglia or astrocytes that have important metabolic functions between neurons and microglia. And here you see the various degrees of elevation of numbers and of astrocytes, and here microglia in the brains from these patients in different areas, the cerebellum here, anterior cingulate gyrus and the middle frontal gyrus, which are important areas in autism. And along that, with that, we found a pattern of increased cytokines and chemokines in these different brain regions. Here is IL-6, an important pro-inflammatory cytokine, but also TGF-beta and others that are anti-inflammatory. So it wasn't just pro-inflammatory, but we also found evidence for anti-inflammatory cytokines. We also found a similar pattern in the spinal fluid. And as you know, the spinal fluid is a specialized material that is made from blood, specifically bathing the nervous system, and can be obtained by a spinal tap. And we were fortunate to be able to obtain that spinal fluid, and we're then able to show that there's a change in the cytokine pattern. Now here's a diagram, or our initial look at this problem suggested to us that microglial activation might be self-perpetuating because it's present at all ages. We thought that the expression of TNF-alpha, for example, a pro-inflammatory cytokine, would stimulate astrocytes, and that the astrocytes would then produce increase in glutamate, glutamate being a major excitatory neurotransmitter, and then subsequently glutamate would perpetuate microglial activation. And these functions are well known. The question is whether the genetic influences might be affecting the immune system as well as the GI tract and the CNS simultaneously. This is not a entirely unreasonable outlook, but it's not the only way to look at the problem. This is another view we had of neuroinflammation. The question was, is it beneficial or harmful? There has to be a critical balance in everything in life, I suppose, particularly in the nervous system, where, and many other systems, where a little bit of imbalance can probably be tolerated, but too much imbalance tips the scale and may affect normal development, and not only development but ongoing problems. So this is a picture of the cells. We found an increase in GFAP, glial fibrillary associated protein, showing astrocytes increased in autism, as well as microglia stained here with HLA. Now I'd like to shift to what I think is a very important review of the interactions among these three important cells that we've mentioned. And this is a nice article by Zaidan Chulia and the group from Porto Alegre in Brazil in Neuroscience and Biobehavioral Reviews. They talk a lot about the functions of these three important cell groups: the astroglia, that is the astrocytes, oligodendroglia, and microglia. And as you know, oligodendroglia are the main cells that form myelin, or the white matter of the brain, and microglia we've mentioned, and astroglia. These are all important to neurons, and of course neurons are the other important group that might be included here. But astroglia are important for the genesis of neurons. They're important for the formation of synapses and so on, and a lot of metabolic functions in the brain. And of course microglia are important for synaptogenesis, the formation of synapses, and for the networks that synapses form. They're very important for a process we call pruning. Once synapses are formed, they need to be pruned, as if, like you would prune a tree in order to help it grow. Synapses and networks need this pruning process, and microglia play a big role in that. They secrete important trophic factors, and they modulate synaptic transmission and neurogenesis. Here they have an interesting flowchart of the actions of astrocytes, showing their importance for remodeling and restoration, and for homeostasis, metabolic homeostasis. Microglia may be resting, and when classically activated by an injury, become neurotoxic, producing IL-1 beta, TNF-alpha. They're a potent source of reactive oxygen species, oxygen needing antioxidants, and activated microglia of the alternative type that we've already talked about are neuroprotective. They produce anti-inflammatory cytokines, and they produce neurotrophic factors such as insulin growth factor 1. So the picture becomes much more complex as time goes along. And of course, like all things complex, they stir a lot of ideas about different approaches, and we'll talk about that shortly. Here is the flow diagram produced by the Brazilian group, in which they suggest that gene-environment interactions may play a role upon a genetic basis and affect brain development in the fetus during pregnancy. They emphasize here that abnormal epigenetic gene regulation may play a role in this. And what I'm going to suggest is that it's the epigenetic regulation that may help us to understand all the many abnormalities in the immune system in autism and perhaps in other areas as well. This is associated with increase in oxidative stress, dysregulation of mRNAs, and leading to toxicity and neural insult. And these ultimately lead to the dysregulation of neuron-glia interactions among microglia, astrocytes, and neurons. This is from a paper by Rezi and Mali in 1999. This group and others demonstrated that microglia are very active during the second and third trimester of pregnancy. In other words, there's an increase in their number and their activation, and their functions are very important during brain development. And their functions during brain development may be different than they are later postnatally. But here you can see they originate in the next to the ventricles in the brain, in the subventricular zone, and they travel along radioglial guides to get through the cortex, and they take up stations in various areas in their travels along here. They're responsible for generating blood vessels in the brain, and they probably play an important role in the actual formation of the cortex. They are activated during this period, very active, and the question arises whether their activation at that point, sometime during pregnancy, may actually become arrested in terms of reprogramming and preventing them from assuming or participating in a more mature role in the nervous system. So what I'm suggesting is the idea is that their activation may be persistent because of an arrest in their development. This is from a nice article by Elaine Shao from Caltech last year, in which she outlines many of the things we've talked about, the maternal antibodies and effects during gestation, and I would suggest that maternal antibodies persist. We measured them in mothers as long as 18 years after birth of their child. So we know that they're long-lived antibodies, and what stimulates their ongoing production is not at all clear. Here are the neuroimmune abnormalities and the peripheral immune abnormalities we talked about, which have been measured, of course, in childhood and after that, but we really don't know when their onset is, and that's a big question as to when these originate. It would be very difficult to tell, but I think there are some ways of looking at that. I'd like to shift again, and let's talk about treatment or immunomodulation. And the question arises, could we change any of these findings in the brain? Lots of people have tried, There hasn't been very much systematic investigation in the way of controlled trials. Most of it is anecdotal, and there are many reports of positive effects, but nothing that we can really depend on in terms of hard data. IVIG, or intravenous immunoglobulin, has been used for a number of years with the idea that immunoglobulins tend to be decreased, particularly certain types of them, and improvements in autism have been reported, but as I said, it hasn't been systematically studied. Prednisone is a common treatment for immune abnormalities, usually thought of as immune suppression because it suppresses immune responses in autoimmune diseases. And many children have received this, and again, anecdotal reports suggest that it is effective, although it can't be used long-term usually. It has many side effects, and it's not at all clear what it's doing in autism, because people who have treated have not measured various immune functions, which needs to be done. Minocycline is a form of tetracycline used for treatment of acne, and was used in a small trial at NIH, an open label trial in children with autism, and was shown not to have a clinical effect. Now, the reasoning behind minocycline is that it is known to suppress microglial activation, so it made good sense to try to do this, but it did not appear to be effective. Pioglitazone is a drug for diabetes. It has some anti-inflammatory effects, and there is a paper on that. Infliximab, or Remicade, affects TNF-alpha and is a treatment for rheumatoid arthritis. Experimental models, I think, could be very helpful in testing therapies. The valproic acid mouse model has been used widely. There are Fragile X mice, and of course a third of Fragile X patients have autism. Bone marrow transplant would be interesting because it's been demonstrated that you can correct microglial abnormalities in animals by transplanting bone marrow from a normal mouse to an abnormal mouse. Stem cells are a theoretical possibility and have been actually used clinically in some places. So the question is, do we suppress the immune system, stimulate it, or regulate it? And this is an important question, and one that we are going to have to answer eventually. Suppression is one way of approaching it. Stimulating, I think some people have tried as well with various vitamins and cofactors and so on. But it may be that regulation is more important. Now, we've been interested in fever and the fever response in children with autism, and you probably know that probably at least 20 or 25% of children with autism have a easily recognizable improvement in autistic features during illness and fever. When we actually looked at this, Laura Curran and our group in 2007, we observed this directly at the time of fever. Compared to controls, we found that 83% of our children improved in at least one domain on the aberrant behavior checklist during fever. The most common improvement was decrease in stereotypy, but we were most interesting was a decrease in inappropriate speech. The children had, of course, lethargy, increased lethargy during fever, but also had decreased irritability and hyperactivity. What would be happening during fever that would lead to such dramatic improvement in some of these children? Well, we know many of the well-established findings in autism. Here's just one list. I'm sure everybody could make another list and a much longer list, but here are some that are very interesting. Jill James and her group have demonstrated the importance of oxidative stress, low glutathione levels, and enhanced lipid peroxidation. Of course, neuroinflammation we've talked about, elevated nitric oxide synthesis, impaired mitochondrial function, and energy generation. NRF2, nuclear-related factor 2, is an important transcription factor that transcribes gene information, and it can be seen as one of the epigenetic regulators. And NRF2 levels are depressed, and its genes under its control have lower expression. We've been interested in a drug, actually a nutraceutical, you might say, called sulforaphane, an important product in broccoli, and most concentrated in broccoli sprouts 3 days old. We've been running a clinical trial of this in young men with autism, and the thinking was that sulforaphane may act in some ways like fever. And by this, I mean there are 3 pathways here, here, and here through which sulforaphane can act. One is by stimulating heat shock proteins, an important defense mechanism in all cells. And heat shock proteins are also stimulated during fever. Sulforaphane also regulates NF-kappa B. Remember we mentioned that this was an important gene in the immune system and its expression. And this pathway is the NRF2 pathway, in which it translocates, when stimulated, translocates to the nucleus and affects gene transcription. So these are 3 important metabolic pathways that may be affected in fever, and we're hoping that molecules like sulforaphane that are non-toxic and from nature might be able to simulate some of the important functions of beneficial functions of fever in autism. Here we are back at our elephant, and voila, one area we haven't stressed is mitochondria, and this is an area that many of us have been interested in autism, because the mitochondria are so important for energy regulation in cells and appear to be deficient in functioning in many children with autism, maybe as many as 20% or more, depending on how you test it and look at it. This is a very important recent paper from Giulivi and Napoli et al. And they looked at the immune response in granulocytes, as polymorphonuclear leukocytes, from children with autism. And they showed that mitochondrial function is significantly decreased. But not only that, they found that NRF2 gene expression was reduced. This is a very important finding, and I'm sure will be replicated in larger groups. This is a small sample they looked at, but it was very carefully done. And these are all mitochondrial functions here: the resting oxygen uptake, NADH, succinate oxidase, and ATPase. So the point is, one of the points is that there are many different ways of looking at immune function in autism. So what causes neuroinflammation and immune dysregulation? Which is it, neuroinflammation or immune dysregulation? I would say it's both, but it's primarily immune dysregulation. So this is a quiz. This is your quiz. Is it exogenous? Is it from infections, valproic acid, environmental factors in autism that cause this immune dysregulation? Is it autoimmune activation? C, is it altered immune gene expression and brain development? Or D, is it cellular metabolic dysfunction? Or E, all of the above? It is indeed all of the above, and it is our challenge to continue sorting this out and trying to develop understanding and rational treatments. And hopefully in time we will catch up with Jabba the Hutt and begin to make progress with Princess Leia's help. I want to thank my collaborators over the years, many wonderful people from Tennessee and Baltimore and at MGH in Boston, and now at the University of Massachusetts. I'm grateful to all of them and to all of the people who have, and foundations and who have supported my research. I thank you very much for your attention, and now I hope we have time for questions.
We do have a lot of questions. The first one that a lot of people had questions about was testing. They're wondering about testing that's available now to look at inflammation and what treatment might be available if it's indicated. So you talked about some of that, but I wonder if there are any specific tests that parents might be thinking about.
Tough question. It's possible to do testing at commercial labs of different elements of the immune system. I think probably the most useful one are the immunoglobulins, IVIG, and the subsets. You can also get testing of T-cell subsets. The problem is what to do with the information. I think the most important thing is to have someone you're working with who understands the immune system, preferably somebody with a background in immunology, an immunologist or a person who specializes in this type of thing, and who understands how to interpret the tests. Unfortunately, there's at this point, there's not a commercial test available for maternal antibodies, but I think there is going to be in the not too distant future, and I think that'll be useful, although we don't yet know what the real meaning of it is. But I think that's going to become clear in a short time.
So this question is asking, this parent is asking about seizure disorders. He's saying his son takes valproic acid
For seizures, valproic acid is a well-established treatment. It's very safe, and it needs to be monitored with lab tests periodically for the level of the drug and for its potential effects on the liver and so on.
Okay, but no, is there any link to it exacerbating any of the symptoms associated with autism?
Not that I'm aware of. It is, of course, prenatally it has toxic effects on the developing brain, but and it has actually been used by some practitioners in early treatment of seizures in autism with some suggestion of benefit.
Okay, the next one. My daughter regressed after viral illness. She might have immune dysregulation. She has responded well to treatments that we've used. Do you have thoughts about treatments like IVIG that you discussed?
She regresses after illnesses, with illnesses.
Yes, the parent saying she regressed after a viral illness. Of course, you can't give medical advice, but have you observed IVIG helping individuals who might fit that profile?
Mm-hmm. In that situation, I mean, it's fine to look at immune functions, of course, but children who regress with illness, you should think about mitochondrial functions, because we know they're deficient in many children with autism, and it's been suggested that there may be a link between regression and illness, and particularly in children with autism who have mitochondrial dysfunction. So I would look for someone, a metabolic expert or a metabolic geneticist, or a pediatric neurologist, any number of people who might be willing to look at the question of mitochondrial dysfunction, because if you detect that, it's something that is potentially treatable with specific vitamins aimed at mitochondrial function.
Okay, the next one, this parent's asking about sleep. You had mentioned sleep during your talk, and the parent is asking about strategies to regulate sleep patterns in autism, since a lot of children have insomnia.
Oh, that's a talk in itself. That is a real challenge. Children with autism have particular difficulty maintaining sleep. They also have trouble getting to sleep, but maintaining sleep is equally a greater problem. And it probably has to do with regulation of sleep in important areas in the brainstem. Melatonin is very helpful for getting a child to sleep, and now there are long-acting preparations of melatonin that help to maintain sleep, if you haven't considered that. There are other drugs, of course, that can be used for sleep, but I think there are a lot of things we can do for sleep hygiene, such as sleep scheduling and decreasing light stimulation, particularly computer screens and TV in the evening prior to sleep, because those things depress one's own production of melatonin and inhibit sleep. So there are many different factors involved in sleep and many ways to look at it. But again, I would commend you to sleep specialists, many of whom have a lot of experience with autism.
Okay, another question. They're referring to Bob Navio's work at University of California, San Diego. Are you familiar with his work?
I am indeed.
Oh, okay, great. This person's asking, Bob Navio is researching foundational mitochondrial dysregulation via, I believe it's suramin, looking at abnormal ATP production. Do you have any thoughts on that research?
Yes, I think it's wonderful. I think it's very promising, and I would follow it closely. I don't know if it's ready for prime time yet, but I think that it's well in the works, and it's a very very reputable, and I would encourage you to stay with it.
So this is a curious question. This person's asking, what about maternal fetal antibodies might explain higher numbers of males with autism?
Good question.
Yeah, it's a great one. I don't know if it's an easy one to answer.
Good question. I don't think I have the answer to that, but there are many reasons why males are more effective, not the least of which is the fact that X chromosomes are probably protective, and of course males only have one. But there are a lot of biological processes that affect males more than females, and I can't say specifically about maternal antibodies and how that would work. And as far as I know, the offspring whose mothers have maternal antibodies have the same ratio of male to female. So you would think that if it wasn't discriminatory in that regard, they would be equal, but they're not. So I'm sorry, I don't have the answer to that.
It's an interesting question. How about this one? They're asking about thyroid, about issues surrounding the thyroid with autism. Have you got any thoughts on those connections?
Well, the thyroid prenatally in mothers, or well, and we know prenatally that hypothyroidism has long been suggested as having an important role in brain development, and the question is whether it may play a role in autism as well. I always check thyroid in children with autism, but it's unusual to find hypothyroidism, and I certainly wouldn't advocate doing more than checking for it and only treating in situations where it's absolutely indicated.
Okay, we're almost out of time, but this is an interesting question. This person is asking, does serious physical injury or trauma cause immune dysfunction, autoimmune disorder, or brain inflammation? My son had a traumatic incident, and he regressed and quickly lost skills.
I would say yes. There's no question that trauma induces increased immune activity. It's actually a normal response to trauma. And yes, I think that it could induce regression. The question is, there are many aspects of that question, whether the child had signs of autism before the injury. Injury to specific areas of the brain can produce autism. I've seen that, and infections as well, if they particularly affect the temporal lobe and frontal lobes.
Okay, I think we've got time for just one last question here. This person's saying hello to you. Okay, this is someone you must know. Yeah, sorry. Okay, so here's the last question. Does neuroinflammation and immune dysregulation, do they share a link with methylation dysfunction?
I think they do. Well, everything is related one way or another, but I think that probably they do affect. They certainly do affect epigenetic regulation, and probably through methylation as well.
All right. Well, thank you so much, Dr. Zimmerman. This has been a great talk today. We really enjoyed it. I wanted to remind everybody still on the call, we sent out a link prior to the call that had the link to the handouts. So if you're looking for the slides, that was in the email that we sent this morning. We also provided a link to our ariconference.com site, which is where we will be posting the video later this week. And we had a link for the quiz if you're hoping to take the quiz after you attend the webinar. I want to invite everybody. Later this month we'll have Dr. Suruchi Chandra speaking. She'll be talking about anxiety in autism. So if you'd like to attend that, please check out our website at ariconference.com and get registered. Space is limited, so that's on June 25th, and we'd love to see you there. We appreciate everybody's time, and again, Dr. Zimmerman, thank you so much, and we look forward to seeing everybody online again soon.
And thank you.